Good morning to all. Today's point of discussion is galvanic cell or voltaic cell. Now remember, galvanic and voltaic are the same assembly. So word name galvanic cell is in the honor of Luigi Galvani, who first of all prepared the electrochemical cell. Later Alessandro Volta modified that electrochemical cell, thus it is also called as voltaic cell. Now remember what galvanic cell or voltaic cell is exactly. Remember, this cell consists of two separate concentric beakers in which two electrolyte solutions are taken, and a metal having the same ion to that of electrolyte is immersed in the electrolyte solution, and two electrolytes are to be connected indirectly by means of the salt bridge, and metal electrodes are connected by means of conducting metallic wire. So this assembly is a voltaic cell or galvanic cell, and one of the finest examples of this is a Daniell cell. Now let us explain Daniell cell. So first of all, how to construct a Daniell cell. So first of all, construction. Remember, Daniell cell consists of two separate concentric beakers which can be drawn as, so these are the two beakers. In one of the beakers, zinc sulfate solution is present, that is ZnSO4, and zinc electrode is immersed in it. And in another beaker, copper sulfate solution is taken and copper electrode is immersed in that. And these two solutions are connected indirectly by means of the salt bridge. So this is the salt bridge. Later we will explain what is exactly a salt bridge. And these two electrodes are to be connected by means of a metallic wire which happens to be good conductor. Now this is a CuSO4 solution, that is copper sulfate solution, say 1 molar. And this is a copper rod which is immersed in it, and this is zinc sulfate solution, that is ZnSO4 solution, and this is the zinc rod which is immersed in it, and this is a salt bridge. Now this is the simple construction of what Daniell cell. So later we will explain what is exactly salt bridge and what are the functions of it. Now we can't draw the figures at every time, thus we need to take care of, we need to use simple notations in order to express the cells, and that simple notation is formulation of cell. In next coming lectures we will discuss about the formulation, how to represent the galvanic cell. So first of all, the point is Daniell cell. So how to represent this, formulation of cell, that is Daniell cell. Now in brief, one thing should be noted. In left hand side we need to mention oxidation half. Electrode at which oxidation takes place is needed to be mentioned at left hand side, and at extreme right reduction half cell. And single vertical line is drawn in between metal electrode and solution around it, and which represent, single vertical line represent direct contact. And in order to indicate indirect contact between two electrolyte solution, we need to use double vertical lines. And the concentrations, if known, those should be mentioned just below the electrolyte. Now, formulation of this Daniell cell, oxidation takes place at zinc, therefore it should be written at extreme left. So it is in contact with what? ZnSO4 solution, say 1 molar. One can write ions as well. Instead of writing ZnSO4, one can write Zn2 plus also. And ZnSO4 solution is in indirect contact with CuSO4 solution. Now say it is also of 1 molar. Instead of writing CuSO4, one can write Cu2 plus also. And in CuSO4, a copper rod is immersed, therefore single vertical line Cu. So this suggests the formulation of the cell, that is the representation of cell. Now we need to take care of working of cell. Working of cell. I mean how cell works. First big thing is zinc is electropositive as compared to copper, first big thing. Secondly, its solution pressure, I mean solution pressure of zinc is greater than that of osmotic pressure, and such type of metals which have high value of solution pressure are capable of dispersing ions in the solution and electrons at electrode, that is called as de-electronation. So at zinc electrode exactly what happens? At zinc electrode, oxidation of zinc takes place. So zinc oxidizes, producing Zn2 plus ions, and electrons are liberated. So this is the oxidation reaction. As oxidation is taking place at zinc electrode, it is said to be anode. Another big thing is Zn2 plus ions are entering the solution and electrons at electrode. Thus zinc electrode carry negative charge as well. Now, so if Zn2 plus ions are entering the solution, then electrical neutrality of solution must get disturbed, but it is not to be. Now salt bridge, what salt bridge does? Salt bridge disperses the negative ions and hence maintains the electrical neutrality. Now at other electrode, I mean at copper electrode, what happens? At copper electrode, copper ions are there. Remember, copper is a metal whose osmotic pressure is greater than that of solution pressure, and such type of metals are capable of gaining electron and get deposited at electrode. I mean metal get deposited at electrode and electrons go into the solution. Thus Cu2 plus ions whatever present at this area gain the electrons lost in the process of oxidation and undergo reduction. So remember, this is the reduction reaction taking place at copper electrode. Now as reduction taking place, and remember its osmotic pressure is greater than the solution pressure and electrons are going inside the solution and metal get deposited at electrode. Thus this electrode carry positive charge and the process of reduction. Now when we add these two reactions, we get the net cell reaction. Now net cell reaction is, if we add oxidation and reduction of reaction, electron get cancelled. So what remain? Zn plus Cu2 plus gives Zn2 plus Cu, and we are using 1 molar CuSO4 and 1 molar ZnSO4. Therefore these concentrations can be written as, now as Cu2 plus ions getting reduced from this side, now positive charges in the solution undergo and it decreases. Now how the electrical neutrality is maintained right here? Now again salt bridge play vital role. Salt bridge releases the positively charged ions from it inside the solution and maintains electrical neutrality. So this is the redox reaction. Now conclusions, exactly what happens? Zinc oxidizes, dispersing Zn2 plus, therefore size of zinc electrode decreases, and on other hand there is a reduction of copper ions and deposition of copper at electrode takes place, thus size of copper rod seems to get increased, and the potential developed by this cell is found to be 1.1 volt. So it was the working of Daniell cell, which happens to be voltaic or galvanic cell. Now very crucial thing, we used salt bridge to connect two electrolytes, I mean two half cells. Now question arises, what is salt bridge exactly? Now salt bridge is an inverted U-shaped glass tube which can be mentioned as, so this is supposed to be the beaker in order to feed the salt bridge. Now let us explain what salt bridge is. It is inverted U-shaped glass tube which is filled by the saturated solution of KCl and agar gel. Now saturated KCl is filled in it and agar slurry, remember 5% agar gel is added to that solution. What is the purpose of agar gel? Agar gel on cooling solidifies the solution, I mean saturated KCl solution, thus it will not come into the solutions when we invert the salt bridge. And here cotton plugging is there which can be termed as glass wool plugs. And remember, instead of using KCl, one can use KNO3 or NH4NO3 also, but one can't use NaCl. So in salt bridge only those electrolytes are to be used whose both ions, I mean cation and anion, should have nearly same transport number. Then and only then that electrolyte will work. NaCl cannot be used right here. If we use NaCl, what will happen? Actually Na plus and Cl minus ions have different transport numbers. Thus NaCl solution when incorporated in salt bridge will not work for sure. Therefore only such type of electrolytes are used in the salt bridge, I mean KCl, KNO3 or NH4NO3. Now why we are using the salt bridge? So what are the functions of salt bridge? Now remember, very first big thing, very first function to be explained is salt bridge connect two half cells and complete the electric circuit. So it provide electric contact. So very first thing, it provides electric contact and complete the electric circuit. So this is the very first function of it. Second thing is it doesn't allow two electrolytes to mix. So consider this again, I mean Daniell cell. In this, in one beaker we are using zinc sulfate solution and in other beaker we are using copper sulfate solution. Salt bridge doesn't allow these two solutions to mix. I mean it avoid mixing of electrolytes. It is important function. It avoids mixing of electrolyte solutions. And third important thing is during the working of cell, oxidation and reduction takes place, and due to which charges, I mean solution would acquire certain charge. So electrical neutrality is prevented by salt bridge. So salt bridge plays vital role in prevention of electric neutrality. So it maintains electrical neutrality, and thus cell works. Remember, if salt bridge fails, then cell stop producing current and voltage of cell get down. Thank you very much.